Hello, and welcome to the next in our series of Plant Presents webinar. Today, we are recognizing the publication of a special focus issue by the Plant Cell with a focus on plant biotic interactions. We have three speakers. Our host is Yangnan Gu, a Plant Cell reviewing editor, and our moderator is Tatsuya Nobori, a Plant Cell assistant features editor. The focus issue is now online. You can find a recording of this and other webinars on our YouTube channel. I'd like to give a special thank you to ASPB member attendees. If you're not a member and you'd like to support great science, please use the code PRESENTS10 to get a 10% discount off your membership dues. If you're having technical problems, please email me, mwilliams@aspb.org, and try reconnecting maybe. If you have questions for the speakers, please type them into the questions box, and Tatsuya will read them out after each presentation. And now I will turn this back over to Yangnan, who will tell us a bit more about the focus issue.
Thanks, Mary. All right. Good morning, good afternoon, good evening, everyone across the world. Welcome to the webinar, and I am Yangnan Gu. I'm one of the Plant Cell editors who also participate in organizing this focus issue for Plant Biotic Interactions. So the reason that we organize this special focus issue is because hopefully everyone has been aware of that our Plant Biotic Interaction has made series of major breakthrough in the last few years, beginning with characterization of the exciting first near atomic structure of the plant resistosome, and for subsequent functional characterization of NLR proteins which form those resistosome, and showing that they could form those calcium permeable channels and also cleave an AD to generate signals to activate the effector triggered immunity. We are also beginning to understand how different diverse immune signaling pathways, including pattern-triggered immunity and effector-triggered immunity, which we use to separate them apart, and how they actually integrated in the plant cell to activate immune responses, and those two are actually interdependent. More than that, other exciting advances, including that we are starting to understanding how the small RNAs are translocated interkingdomly between the plant and the pathogens, and a clear understanding of how salicylic acid is perceived and how their receptor, including MPR proteins, their structures, including a few days ago, the MPR1 structure was out on Nature, and how the immune signals can move systematically through the plant, and what are the signals can activate systemic acquired resistance. So these are all long sought after questions in the field, and I'm just listing a few of them. So in this focused issue, we collected 8 review articles and 14 original research articles to exemplify this rapid pace of discovery and highlight the breadth of the research being pursued. So I just want to take a few minutes to briefly advertise our focused issue and go through those exciting review papers that we collected, including 3 papers that are talking about resistosome and the NLR receptors. One from Gigi's group talking about 30 years of advances of this original zigzag model to our current understanding of the plant immune system, and also from Jim Parker's and Xin Li's lab about how the T receptor is immune signaling, and also a large-scale biology paper about the origin and evolution of plant resistosome. We also have 3 papers that cover comprehensively of different topics about the plant interaction with insects, with virus, and also their interaction with symbiotic microbials. Also a few exciting topics about plant microbial interaction with the cell wall, and the recent advances in the calcium channel, their function in the plant immunity, and the emerging role of liquid-liquid phase separation of biomolecular condensates in activating immune responses. Of course, as I talked about, we also have collected 14 original articles, which covers a broad spectrum of topics, including NR receptors, effector release mechanism, the role of RNase, E3 ligase, nucleoporins, and kinases in activating immune responses. I should emphasize that actually many of those papers and reviews are open access. You can read them without a subscription. So I highly encourage you to read this focus issue. So for today, we selected 3 out of the 14 excellent review articles and let the authors of those articles to present their own research. This part will be coordinated by our excellent assistant feature editors, Tatsuya Nobori. So Tatsuya is a long-term human, I think it's long-term human frontier science program long-term fellow. He is currently working with Joe Acker in Salk as a postdoc. He's studying the plant microbial interactions at single cell levels and also exploring the epigenomic features during the plant microbial interactions. So Tatsuya, take it away.
Yeah, thank you, Yanan, and thank you, Mary. I'm very excited to be moderating 3 talks in this webinar. So now I'm going to introduce our first speaker, Peipei Qi. She is a PhD student in the lab of Xiao Yu at the Huazhong Agricultural University in China. She studied her PhD in 2020. Before that, she studied in the laboratory of Bo Li at the same university. She studies Rhabdostonia effector that interferes with plant immunity using Arabidopsis in tomato models. And today, she will be highlighting her recent plant cell paper entitled as Rhabdostonia serum effector targets TGA transcription factors to subvert salicylic acid signaling module. So welcome, Peipei.
Okay, thank you very much. Okay, can you see my screen?
Looks good.
Okay, thank you very much. Hello, I'm Peipei, a PhD student from Huazhong Agricultural University, and it's my great honor to present our work here today. Today, I will talk about Rhabdostonia solanacearum effector targets TGA transcription factors to subvert salicylic acid signaling. First of all, the bacterial pathogen Rhabdostonia solanacearum causes plant bacterial wilt and leads to great economic losses every year due to the wide range of hosts, genetically diverse, and the lack of stable resistance gene. During infection, the bacterial mainly use cell wall degradation enzymes, X-polysaccharide, and TAPS effectors for its colonization and cause plant wilt and death. Like other bacterial pathogens, the main virulence factor of Rhabdostonia is TAPS effectors. To date, many kinds of Rhabdostonia effectors interfering with plant immunity or metabolism have been identified. For example, the TAPS effector RIP-AY and RIP-I. However, how individual effectors interfering with plant immunity, especially in transcriptional reprogramming, remains elusive. So, in our research, to identify the TAPS effectors of Rhabdostonia interfering with plant immunity expression, we screened a batch of plant nuclear localized TAPS effectors of Rhabdostonia GML 1000. And we found the core receptor effector RIP-AB could significantly suppress the Rhabdostonia pump F18 induced FRQ1 and SA analog LA induced TR1 induction in Arabidopsis protoplasts. In addition, the FLAG22 induced ROS burst was greatly suppressed in tobacco. So these results suggest that RIP-AB may influence plant SA and ROS signaling. So next, we constructed RIP-AB overexpression plants. And consistent with the transient assay results, we found the immune marker genes induction were significantly decreased in RIP-AB overexpression plants. And the rust burst as well as callus deposition induced by RIP-18 were also compromised in RIP-AB transgenic plants. So these results confirmed that RIP-AB could suppress plant immune response. So next, we checked the whether RIP-AB influenced plant resistance to Ralstonia. We found RIP-AB overexpression plants show the enhanced susceptibility and supported more JAMA 1000 growth. So this result indicated that RIP-AB expression could impair plant resistance to Ralstonia. To check the importance of RIP-AB to Ralstonia's virulence, we generated RIP-AB deletion and complementation strains, and we found the RIP-AB deletion strains completely lost virulence and could barely grow in tomato moneymaker, while the complementation strains could restore its virulence. So RIP-AB is required for Ralstonia's virulence. So in order to explore the mechanisms of RIP-AB interfere with plant immunity, we performed IP mass spec in tobacco to identify RIP-AB target protein in plants. TJ2 transcription factors was among the targets. There are 10 TJs in Arabidopsis, and these were classified into 5 clades, among which the functions of clade 1 and clade 2 in different plant defense pathways has been well reported. Now, the clade 1 members TJ1 and TJ4 regulate the biosynthesis of SAA and the peptidic acid. The clade 2 members TJ256 involves in SAA signaling. So to confirm the interaction of RIP-AB with TJ2 as well as other immune-related TJs, we performed the bimolecular fluorescence complementation and the split luciferase acid, and we found the RIP-AB indeed interacts with TJs in tobacco, and we further confirmed their interaction by protoplast co-IP and in vitro prodown assay. And because RIP-AB interacts with TJs, we checked whether RIP-AB influenced TJ's function in plant immunity. Surprisingly, we found RIP-AB displayed similarly as TJ14 mutant, and displayed compromised FLAG22 induced disease resistance. In addition, TSM induced STAT1 and CBP6 gene expression was also compromised in both these two plants. So this result suggests that the RIP-AB could block TJ1 and TJ4 mediate plant immunity. Same as above, we found the RIP-AB overexpression plants were hypersensitive to RNA, and RIP-AB overexpression plants almost lost the RNA-induced resistance and SAR response, which is with copies TJ256 triple mutant in disease resistance. So this results suggest that the expression of RIP-AB could block the TJ256 related plant immunity. In order to explore the biological relevance of TJ in RIP-AB virus function, we challenged TJ mutants with JAMA 1000 and RIP-AB mutant strains. And we found, in contrast to the significantly weak virulence to wild-type plants, the RIP-AB mutant strains showed enhanced pathogenicity and grow more in TJ mutants. So this result suggests that the TJ factors are the bona fide targets of RIP-AB. To explore the influence of RIP-AB on induced immune gene transcriptional reprogramming, we performed RNA-seq in wild-type and RIP-AB transgenic plants treated with RNA, and we found the global gene expression didn't change significantly upon RABAB overexpression, but the RNA-induced differentially expressed genes varied a lot. Most importantly, we found the assay and defense-related genes are suppressed in the presence of RABAB. Moreover, the RABAB significantly suppressed the RNA-induced RBHD induction. And our ROS assay results were also confirmed the results. Since RABAB target TJs, we wonder how RABAB influenced TJ's function in plant. So firstly, we tested the TJ's transcriptional activity in the presence of RABAB with a GAR4 transcription system. And we found RABAB could largely inhibit TJ's transcriptional activity induced by F18 and RNA. And we also checked the influence of RABAB on the binding activity of TJs to the target genes promoter, and we found that RABAB does not influence the TJ binding activity. It has been reported that when the assay level increase, MCR1 binds SA and enters the nucleus to recruit TJ256, then promotes the activation of immune gene transcription. So we checked the influence of RABAB on the RNA-induced interaction of MCR1 with TJ2 and the EDS1 with the bimolecular fluorescence complementation and the Peipei assay. And we found that RABAB does not influence the RNA-induced TJ2 and MCR1 complex formation. Since gene expression requires transcriptional machine and RAPL2 to initiate transcription, so we checked the influence of RABAB on the recruitment of RAPL2 to the TJ target promoter, TJ target genes promoters. And we found that RABAB could inhibit an induced interaction between TJ2 and RAPL2 with the protoplast Peipei assay. And we also found the induced enrichment of RAPL2 to the TJ target promoters was also compromised in the presence of RABAB. So based on the above results, we summarize the working model here. The Rastonia types effector RABAB is secreted by the TAP3 secretion system into the plant cell and translocated into the nucleus. On the one hand, RABAB could interact with transcription factors TJ1 and TJ4 to suppress the PAM-triggered immunity. And the assay peptolic and the assay and the peptolic acid biosynthesis gene expression. On the other hand, RABAB also interacts with TJ256, causing the compromised SA-mediated resistance. So finally, I'd like to thank the fans for supporting our research, and thanks the co-authors Wu Xuehan, Hong Menglin, and the colleagues in our lab for kindly help in our research. And we appreciate Professors Zhang Yuelin, Chen Huilan, Cao Yangrong, and Wang Taotao for sharing these our streams. Okay, that's all my report. Thank you for your attention. Thanks.
Thank you, Peipei, for a great talk. Also, you are right on time. It's great. So we got already several questions, but please, everyone, type your question into the Q&A box so I can ask for you. So first question is from Ron Lee, who asks how big the sample size for you to do the ROS assay. So how many replicates and samples have you used, and how did you see the high variability in the data in the ROS assay?
Oh, sorry, I didn't hear it clearly. Could you please say it again?
Sure. So you showed the ROS assay data, right?
Yeah.
ROS burst, reactive oxygen species. And so this person asks like how the sample size in this ROS assay. So how many replicates or how many samples you have used in your ROS assay?
ROS assay?
Yes.
In our experiments, we usually use 20 to 30 samples to measure the ROS burst.
Okay, yeah, you just answered the question. Thank you. So the next question is from Jason Perez Chavez, who asks, so how do you think your finding could be translated into the real world in the agricultural application? Do you have any thoughts?
Sorry, I can't stop my.
Oh, maybe Mary can help you unshare the screen. There we go. So
Okay.
did you hear the question?
Sorry, could you pardon?
Yeah, sure. So the person asks, how can you translate your finding in this paper into the agricultural application?
Peipei, you can read the question if you click on the Q&A box. You can read it. Maybe that would help.
Sorry, I can't see the questions.
Okay.
Okay, yeah, maybe I'll move on to our next question.
Yeah.
So, all right, so Kensaku Maejima asks, which domain or region of TGAs is the target of RIP-AB?
Okay, in our research, we checked the information. We checked the interaction of RIP-AB with the TGAs N-terminal and C-terminal, and we found RIP-AB interacts with their both. So we think RIP-AB targets the whole TGAs domains.
Mm-hmm. Great. Maybe last question. Niraj Agarwala asks, which Ralstonia strain did you use for your virulence assay, and what type of inoculation strategy have you used for the pathogen assay?
In our research, we use the Ralstonia strain Gemma 1000.
Mm-hmm.
Yeah.
And how did you inoculate the pathogen?
We, in our research, mainly use the root inoculation.
So directly into the soil, or?
Yeah, like this in the soil to the wounded root.
Mm-hmm. Okay, perfect. So I think that's all for this session. So there are still several questions unanswered. So if you can access the Q&A box, you can also answer the questions by yourself. So thank you, Peipei, for your great talk.
Okay, thank you.
All right. So now we'll move on to the next speaker. Next speaker is Yugo Kitazawa. He is an assistant professor at the Laboratory of Clinical Plant Science at the University of Tokyo, Japan. He obtained his PhD from the same university in 2017 and did his postdoc also in the same university until last month. He studies phytoplasma that turns plant flowers into leaf-like structure. And today he will talk about molecular mechanisms underlying this interesting phenomenon, highlighting his recent plant cell publication entitled as "A Phytoplasma Effector Acts as a Ubiquitin-like Mediator Between Floral Matchbox Proteins and Proteasome Shuttle Proteins." So Yugo, please take it away.
Thank you for introducing. Can you see it?
Yes.
Okay. Thank you for introducing. I'm very happy to have this valuable opportunity for presenting our research. Phytoplasmas are a group of low and limited plant pathogenic bacteria. They can infect over 1,000 plant species and induce unique symptoms such as yellowing, proliferation, and failure. My main study is to explore the disease development mechanisms, especially failure. Phytoplasma uses pathogen to induce failure. Pathogen is an effector conserved among phytoplasmas, inducing failure in diverse plants, such as Arabidopsis, petunia, sesame, and sunflower. Pathogen targets mass domain transcription factors, MTF, a regulator of flower development. Pathogen can interact with MTF directly and initiate it to the host proteasome and induce degradation. So my main study in the presentation is how pathogen initiate MTF to the proteasome. Usually, proteasome degrades ubiquitinated proteins. In the first step of degradation, ubiquitin binds to target by the function of three enzymes, E1, 2, 3, and ubiquitinated proteins are recognized by a protein called as RAT23 by the interaction of RAT23 with ubiquitin. Then RAT23 initiate target to the proteasome. This means that ubiquitin is a key factor for proteasomal degradation. In fact, bacterial effectors utilizing host proteasome degrade host proteins in ubiquitin-dependent manner. On the other hand, Dr. McLean showed that pathogen can interact with RAT23 directly. Because RAT23 deficient mutant Arabidopsis thaliana, in the mutants, pathogen cannot induce failure. Therefore, this interaction is considered to be important for the MTF degradation. This is interesting because pathogen can also interact with MTF directly. This suggests a simultaneous interaction of three proteins without ubiquitin. If so, MTF can be initiated to the proteasome via pathogen and RAT23, but independently of ubiquitin. So the purpose of the study is exploring the interactional relationship of pathogen, MTF, RAT23, and if relevant, ubiquitin. For the purpose, we try to detect the ternary protein complex first. Here's the result. For the detection of ternary protein complex, we co-express three proteins in plants. Fused RAT23C, a homologue of Arabidopsis RAT23, and PHIL-OI, phylogen of OI phytoplasma strain, and MICSEP3, a famous E-class MTF. These proteins were co-immunoprecipitated by flag tag. This is result. In the absence of PHIL-OI, CEP3 does not interact with RAT23C, but in the presence of PHIL-OI, CEP3 and PHIL-OI interacts with RAT23, indicating that PHIL-OI induces the formation of ternary protein complex. Moreover, additional interesting results were obtained. First, in the presence of CEP3, ladder-like bands were observed above PHIL-OI. From band size, we speculated this is because ubiquitination of PHIL-OI. On the other hand, such bands were not observed above CEP3. Moreover, in the absence of CEP3, the FIL-OY interacts with RAT23C very slightly. But in the presence of CEP3, FIL-OY interacts with RAT23C stronger, indicating that RAT23 interacts with CEP3 FIL-OY protein complex stronger than FIL-OY. Next, we performed another co-immunoprecipitation assay using frag tag fused CEP3 instead of frag RAT23C. The results were basically same. RAT23C interacts with CEP3 only in the presence of FIL-OY, and ladder-like bands above FIL-OY were observed. On the other hand, regardless of the expression of RAT23C, FIL-OY interacts with CEP3 to the same extent, indicating that CEP3 interacts with FIL-OY RAT23C protein complex as well as FIL-OY. Finally, we confirm that FIL-OY in the protein complex is ubiquitinated. By the detection using anti-ubiquitin antibody, FIL-OY monomer was not detected, but the bands above FIL-OY were detected, indicating these were from ubiquitinated FIL-OY. From this result, we speculated the model of protein complex formation. We detected ternary protein complex, and RAT23C interact with FIL-OY weakly, but strongly with CEP3 FIL-OY complex. This indicates that FIL-OY interacts with host protein sequentially, that is, first with CEP3, and then RAT23C. Moreover, during the process, FIL-OY is ubiquitinated. This is interesting because RAT23 can interact with ubiquitin directly as well as FIL-OY, suggesting the possibility that ubiquitin has some role during the degradation. So we've next studied whether ubiquitination of FIL-OY is important. For the purpose, we created non-ubiquitinated FIL-OY mutant. Usually, ubiquitin binds to lysine residues of proteins. Thus, we created FIL-OY all-KR mutant, in which all lysine residues were replaced to arginine. We confirmed the mutant is not ubiquitinated and studied its activity. However, the mutant induces CEP3 degradation and induced FILD in plants as well as FIL-OY wild type. Thus, we concluded FIL-OY can work without ubiquitination. Finally, we proved that pathogen can directly intermediate MTF and RAT23 interaction without ubiquitin. We performed in vitro co-immunoprecipitation using CEP3 FIL-OY RAT23C expressed in E. coli and purified. Because E. coli does not have ubiquitin, this assay was performed in complete absence of ubiquitin. The results showed CEP3 can interact with RAT23C only in the presence of FIL-OY, indicating the direct interaction of three proteins. That's conclusion. Pathogen induces MTF and RAT23 interaction without ubiquitin. Pathogen interacts with MTF first and then RAT23. From these, the mechanisms of target degradation by pathogen is considered like this. In ever-known bacterial effectors, these effectors induces ubiquitination of target first. Then RAT23 recognizes ubiquitin and initiate target to the proteasome. On the other hand, phylogen can interact with MTF directly and then RAT23. And RAT23 initiate MTF to the proteasome independently of ubiquitin. In this process, phylogen can act as a functionally ubiquitin-like mediator in that it connects target to the RAT23 instead of ubiquitin. This is quite unique bacterial effector function. So that's all of my presentation, but we have concluded, included much more topics in our paper. So if you're interested, we are happy if you read it. So that's all. Thank you for listening.
Thank you, Hugo, for a very interesting presentation. So we have one question from Sir Houston, who asks, is phylogen ubiquitinated and interact with RAD23? If phylogen is ubiquitinated and interacts with RAD23, how it is preventing its own degradation by the proteasome?
Yes, it's a very important, but we don't have the answer. So yes, actually we speculated that this ubiquitination is a degradation signal, but yes, actually ubiquitination field might not induce its proteasomal degradation. Can you see the slide?
Not now.
Sorry. Okay, yes, I don't have the answer, but this is very important question.
So related to this, if you use the ubiquitinated, like the wild type phylogen, do you see any difference in the result of the formation of the ternary complex?
Sorry, pardon?
So you use the mutated, the non-ubiquitinated version of phylogen in your experiment to prove that ubiquitination is not required for the phylogen's function. And what if, so I wonder if you use the wild type phylogen, what would happen?
For this slide? Sorry, can you see
We
this
still,
slide?
no, we still don't see your slides somehow.
Okay, yeah. Okay.
Okay, so now I think you can, yeah.
Sorry. This is for the results of the previous question, showing non-ubiquitinated phyloid mutants cannot accumulate it in planta. Thus, we concluded ubiquitination phyloid might not induce its proteasomal degradation, but the mechanism is unclear. This is the answer for the previous question. And your new question is maybe the difference of the efficacy.
Yes, I think it's also answered here.
Yes, it's also very interesting, but we can't measure the difference precisely. Yes, it's future work.
Okay, so also related to this question, Sabham Agarwal asks, how did you produce this old KR mutant?
Producing method.
Yes.
It's a mutation. Yes.
So you just synthesized or?
Yes, synthesized.
Mhm.
Yes.
Okay. So maybe another question from my side. So I thought that the sequential binding is interesting. So is it advantageous for phylogen to first bind to the target, not to the proteasome complex?
Yes. No clear answer, but we speculated only. In nature, phylogen, MTF, and RAT23 exist in different cells or plants because phytoplasma is phloem limited. Phylogen may be also phloem limited. Like this. And MTF exists for meristem cells. Thus, phylogen may be moved to floral meristem cells and interact with MTF. During the movement, phylogen must pass through other cells. RAT23 exists, but MTF not exist. Thus, for minimizing the interaction with RAT23 in the absence of MTF, it's for the efficient movement of phylogen. This is my speculated model.
Thank you. Yeah, that makes sense. Yeah, how smart. Okay, so I think we have to move on to the next speaker. So thank you, Yugo, for your very nice presentation. There are still several questions in the Q&A box, so you may check these out and answer those questions for yourself.
Yes, thank you very much.
Okay, so our last speaker today is Margo Raffeiner. She is a postdoc in the Houston lab at the ZMBP in Tübingen, Germany, where she studies selective autophagy during plant-bacterial interactions. And she got her PhD in the laboratory of Friedrich Böhnke at the Leibniz Institute of Vegetable and Ornamental Crops in Grossbeeren in Germany, where she studied Xanthomonas effector that interfere with stomatal defense during pathogen attack. And she will talk about this story today, highlighting her recent plant cell publication entitled as the Xanthomonas type 3 effector ZOP-S stabilizes WARKI40A to regulate defense responses and stomatal immunity in pepper. So please take it away.
So thank you. Can you see my slide?
Yes, and I can hear you.
Okay, perfect. Just a second. Okay. Yeah, thank you very much for the introduction. And actually, I would also like to thank the organizers of this webinar for giving me the opportunity to talk about my work today. So I would like to start my talk with some nice pictures of happily growing plants that surround us, because this is what we usually can observe, since seeing diseased plants is rather the exception than the rule. Nevertheless, today we have already heard two interesting talks about pathogens as Ralstonia and phytoplasma that do overcome the plant immune system to cause disease on susceptible plants such as crops or also ornamental plants. And next to the bacterial pathogens that have been already introduced to us today, there is also a large variety of foliar pathogens that can colonize susceptible host plants. And one example is Xanthomonas campestris pathorevesicatoria, or XCV. So XCV is a gram-negative rod-shaped bacterium that has a hemibiotrophic life cycle. And as all other leaf pathogens, also XCV undergoes an epiphytic growth phase upon the arrival on the leaf surface. But after this generally very short phase, XCV enters the leaf tissue through stomata, where it is able to colonize the apoplast. And whenever XCV encounters a susceptible host plant, it is able to proliferate, and therefore it is the main causal agent of the bacterial spot disease in agronomically very relevant crop plants such as tomato or bell pepper. So depending on the strain, XCV translocates approximately 40 type 3 effectors into the host cell. Many of them are not characterized yet. But however, in the past years and decades, huge efforts were made in the characterization of these effectors to really get a better understanding on how XCV is able to cause disease. So it has been shown for some of the effectors that they do interfere with key immunity or plant immunity components as shown here. And amongst these characterized proteins, we can also find XOP-S or Xanthomonas outer protein S, which is, let's say, the star of today's talk. So when I started my PhD in 2017, not very much was known about this effector protein. The basically only information I had is, okay, it is a translocated effector that is about 34 kilodaltons in size, but there was no similarity to any other known protein. And back then, there is also no host target protein confirmed for this effector. But actually, XOP-S wasn't completely uncharacterized because there was a publication from 2012 from the Ulla Bonas lab that could show that XOP-S indeed is responsible for disease symptom development on susceptible pepper plants. So when pepper plants were infected with an XCV wild type strain, leaves displayed severe disease symptoms, while plants that were infected with a XOP-S deletion strain showed an almost overall alleviation of these disease symptoms. But however, Schulze and colleagues back then did not assess a clear virulence function of this bacterial effector protein, since the bacterial growth of the deletion strain, here depicted in red, was not at all reduced when compared to the XCV wild type strain shown here in green. So this was kind of the starting point of my journey with XOP-S. And since there was such a striking phenotype on the pepper leaves that were infected with an XCV strain that translocates this effector, I really wanted to figure out which role does this effector protein play during a compatible interaction between XCV and its host plant. So since the bacterial infections shown here in this publication from 2012 were performed by syringe infiltration, we thought that this experimental approach might mask a potential role of the XOP-S effector protein as a virulence factor. And this is actually because syringe infiltration bypasses a very important layer of plant defense, which is stomatal immunity. So what you can do when you infiltrate bacteria directly into the apoplast is you can observe or study what is happening at a post-invasive stage of infection that I depicted here. But actually what you can't do is making investigations on what happens at the pre-invasive stage. So this phase that is prior to pathogen entry into the leaf tissue. Starting in 2006, when Maeli Melotto from the Shenyang He's lab was able to show that plants are actually able to actively close their stomata upon recognition of a pathogen, what actually led to the coining of the term stomatal immunity. In the past years, more and more effector proteins, but also toxins that are released by pathogens were identified, mostly from the foliar pathogen Pseudomonas, as shown here, that do play a role in preventing stomatal closure in order to benefit the bacterial entry. So having that said, we decided to try changing the way of how we wanted to introduce bacterial strains into the pepper leaf tissue in order to see whether XOP-S really doesn't have any impact on the virulence of XCV. So that's why I went for dip inoculation, which is a method where pepper leaves are literally dipped into a bacterial suspension, and the bacteria have to find their own way into the leaf tissue. And actually, the results that we got were quite striking. So first of all, we could observe or confirm that indeed XOP-S is responsible for disease symptom development, as shown here. So bacteria lacking the translocation of the XOP-S effector protein do not cause severe disease symptoms on susceptible pepper plants, while XCV strains that do translocate this effector clearly do. But on the other hand, we saw a completely different picture when that was of what was observed before when looking at the bacterial performance. So the XCV delta XOP-S mutant strain grew significantly less when compared to the wild type, or the complementation strain that again is able to introduce XOP-S into the host plant. And these results showed that indeed XOP-S is required for the full virulence of XCV. Actually, this is because we could show that XOP-S is able to prevent stomatal closure that usually happens upon a pathogenic recognition. So when measuring the stomatal aperture of pepper leaf discs that were incubated on different bacterial suspensions, we saw that there was a significant stomatal closure of plants that were incubated on the XCV delta XOP-S mutant strain. But whenever leaf discs were incubated on strains that are able to translocate XOP-S effector protein, these plants were not able anymore to close their stomata. And that's why we identified that XOP-S is able to facilitate the bacterial entry into the leaf tissue. So at this point, of course, we also wanted to identify the underlying mechanisms of this XOP-S virulence function. And that's why we try to identify potential host targets of this protein. So by using a yeast to hybrid screen, we were able to identify a WRT40 transcription factor as interaction partner of XOP-S. And this specific interaction with XOP-S was confirmed also for different WRT40 proteins from different plant species, such as for Nicotiana tabacum and Bentamiana, but also for Arabidopsis and the pepper WRT40A transcription factor. So at this point, I would like to very briefly mention what WRT40 is. This transcription factor belongs to a very large family of transcription factors that have been shown to be involved in the regulation of various biotic and abiotic stress responses, but also in plant growth and development. And for the Arabidopsis WRT40, it has been already shown that this acts as a negative regulator of immune responses because it is able to repress the expression of defense target genes together with the WRT60 and WRT18 transcription factors. But so is this negative regulatory behavior also true for the pepper WRT40A transcription factor that was identified as interactor with XOP-S? So what we did observe when downregulating the WRT40A transcription via virus-induced gene silencing was that these silenced plants exhibited significantly reduced disease symptom development when infected with an XCV wild-type strain. And this reduction in symptom development was also very much in line with an enhanced resistance of these silenced plants towards XCV infection. So these data already indicated that also the pepper WRT40A transcription factor might act as a negative regulator of defense. But we could further confirm these data by showing in different ways, one example just over here, that also the WRT40A transcription factor indeed does repress transcription of defense-related genes. And once we had established the interaction between XOP-S and WRT40, we sought also to connect this interaction with the XOP-S virulence function in repressing pathogen-induced stomatal closure. So again, by silencing WRKI40A in pepper via virus-induced gene silencing and incubating leaf discs from silenced and control plants on different bacterial suspension, we could demonstrate that XOP-S indeed does need WRKI40A to prevent stomatal closure. So let me just lead you through this figure. What you can see here at the left side of the figure are the control plants. So in control plants, we have again the same scenario as I have already showed you before, where plants are able to close their stomata only when they were incubated on bacterial suspension that lacks XOP-S effector protein. However, when silencing pepper WRKI40, we see a totally different picture. So the absence of the WRKI40 transcription factor leads to a complete loss of the XOP-S ability to maintain stomata open, revealing that this effector indeed needs its plant target for its virulence function. So another observation that we made while dissecting the function of WRKI40 and XOP-S and the interaction between them was that WRKI40, as many other transcription factors or proteins in general, was degraded via the 26S proteasome. So this makes totally sense for a negative regulator of defense, since it has to be degraded during a pathogenic attack to allow the expression of defense-related target genes. So this is also true for WRKI40, since we showed that a treatment of Nicotiana bentamiana leaves that were transiently expressing WRKI40 with a proteasome inhibitor, MG132, did lead to accumulation of this transcription factor. And very interestingly, we also saw that the co-expression of the XOP-S effector protein together with WRKI40 led to an even stronger accumulation of this transcription factor than MG132 does. So at this point, we thought, okay, it might be the case that XOP-S is able to repress the expression of defense-related genes by stabilizing this negative regulator of defense. And indeed, we could also prove our hypothesis by performing gas reporter activity assays. So by transiently expressing a gas reporter construct under the control of the PR4 WRKI40 target gene promoter alongside WRKI40, we observed that this expression led to a repression of the PR4-driven gene expression. And this repression was further enhanced by co-expressing the XOP-S effector protein. So I would like to end my talk with part of our current working model that summarizes the main findings of our work. So if a susceptible host plant is infected with an XCV strain that translocates XOP-S into the host, at a very early stage of infection, XOP-S ensures that the plant cannot close its stomata, which allows a much higher number of bacteria to invade the tissue. XCV thus translocates XOP-S into the host cell, and in the nucleus, XOP-S is able to stabilize the WRKI40 transcription factor by preventing its proteasomal degradation. And to further suppress, so this further suppresses the expression of defense-related target genes, leading ultimately to a milder immune response. So last but not least, I would like to thank a couple of people. So first of all, of course, my PhD supervisor, Ricky, because we had like a great teamwork that made this work happen. But I would also like to thank all the collaborators and the DFG funding agency for the financial support of the work. And once again, the organizers of this webinar, and thank you very much for your attention.
Thank you, Margo, for a great presentation. So I have one question. So how does the stabilization of WRKI40 actually prevent stomatal closure? So what happens downstream?
Sorry, can you repeat the question?
So the XOP-S stabilizes WRKI40, right? And this is the cause of the inhibition of stomatal closure. So what do you think the mechanism of this?
So what I think is that phytohormones play a very important role, because WRT40 target genes are also phytohormone related, let's say. And by repression of this transcription factor, so by the stabilization of this transcription factor, I think that genes that are involved in the regulation of stomatal aperture are not able to, of stomatal closure, I'm sorry, are not able to be expressed at this moment. So this is what, yeah, I think is the reason, or one of the reasons.
Okay, yeah, thank you. So we got one question. Oh, we are many questions are flowing now. All right. So one quick question is from Alex Costa, who asks, after the infection, when did you look at the stomatal aperture? So how many hours after infection?
So it wasn't a proper infection. Yeah, so it was incubation of the leaf discs on a solution, and we checked about 3 hours after starting the incubation, basically. Yeah, so we had a phase where we let them, so without bacterial suspension, we let them just sit on light, so to maintain like a equal stomatal aperture, and then we started the treatments, and 3 hours after we performed the assay, like we looked under the microscope.
Thank you. So maybe one last question. So Niraj Agawala asks, is the stomata only way through for the Xanthomonas to really get into the plants?
So for the Xanthomonas genus in general, not, but XCV, the strain we use, enters through stomata. So other Xanthomonas species also enter via hydatodes, but this is not the case for the bacterium we are looking at.
Okay, thank you. So we still have many questions, but since it's been already 1 hour since we started,
you can write me an email.
So yeah, so maybe you can screenshot
the
the question box. There are a lot of questions, so
okay.
Which may be helpful.
I will send you the questions, Margo.
Yeah.
So I would like to thank the speakers, Peipei, Yugo, Margo. Really great talks, really, really good. Worth getting up for. Tatsuya, thank you for the expert moderating. Yang Nan, thank for helping to organize this webinar. Thank you, attendees. If you need a certificate of attendance, you'll get an email tomorrow with instructions how to do it. We will be posting this video online early next week. Thank you again for everyone for attending. Marvelous work, really excellent talks. Thank you all so much, and goodbye.
Thank you.
Goodbye.